No, just to say, we've been organizing this special session every year for like 7 or 8 years in the mood of the Nanomedicine Translation Hub. Yeah, exactly. Thank you, Mike. And the idea is just to highlight some innovative project that has a chance to go to the market and not just basic science. I did not check the presentation, but normally they should, all of them, have respected some kind of template, and the template was inspired from the work of the Health Tech Tab, the Translation Advisory Board. And it's a way not just to talk about the technology, not just to talk about the science, but to ask some questions that really put the light on the translational potential of these innovations. I'm super happy to welcome Audrey Nsamela to start. Yes, even with the slides, it would be better. Great. If it works.
Thank you.
I'm super happy for 2 reasons. She could make it despite she will run for the airport, no, for the train station in something like 20 minutes.
Yes.
So don't be late.
Yeah, no.
Respect your time. Yeah, so 10 minutes max.
Quick, yes.
And also because Inside Therapeutics is a brand new ETPN member. You just joined before the conference, and I mean, it looks like you've enjoyed it, and you had a booth, and that's fantastic. So welcome at the ETPN, welcome at this session on the Pitch Me Up session. The floor is yours for 10 minutes.
Thank you. I will start chrono. So hi everyone, can you hear me correctly? Okay, good. I see you all pumped up with coffee, so that's good. You'll be awake. So I'm Audrey. I work at Inside Therapeutics. We're a French-based startup, actually a spin-off company from Helvesis. I don't know if some of you know, where I did my thesis. So working on the topic of lipid nanoparticle synthesis, or more generally, organic nanoparticle synthesis with microfluidics. So we decided to spin off, and I will talk a little bit about the project. And so, yeah, just to mention, the no name is not the name of the platform. It's just that we currently don't have a name yet, and I had to change it due to legal reasons. The name we found was not was already taken. So the mission of the I mean, our mission is actually our vision, is to really pave the way for faster nano-based drug development. So I mean, it's really in the topic of the conference. I really seen also great talks from you, and the idea is to how we can from day one, from the screening stage, provide meet the standards. I mean, I know there is no real standards for nanomedicine. Yeah. Okay. But the idea is how we can control the formulation to have like really repeatable process, repeatable results from the start. And how we want to do that, how we aim to do that, is to provide a scalable and innovative method of nanoparticle formulation using microfluidics. And of course, the targets is to cure cancer, genetic disease, infectious disease, I mean, anything you can think of that you can apply organic nanoparticles. And
just click on the slide, I'm sorry.
No problem.
Click once.
Yeah, okay. So the problem is, okay, there's a weird format. Anyway, so you all know, I mean, I think you know the process of developing a drug. You have to go from the lab scale screening, where you find, you have a lot of parameters to screen, you have the formulation, you have the manufacturing process, you have a lot of things, and you want to find a good candidate based on critical quality attributes. Then when you find a few candidates, you want to have to produce more volumes to go to preclinical and clinical trials. And the next step, once you hopefully go through these stages, you want to increase again the production to distribute your drug to the larger scale. But the problem is that these steps, going from one step to the other, is not so straightforward. You have a lot of bottlenecks, and you have to often change method to go from one stage to the other, and that changes your formulation parameters, your physical, chemical characteristic of your particles. Sorry. So how do we bypass these? How do we bypass these? And unlock these bottlenecks to use the same method across all stages. And so far, we realized that there was no instrument that can go from really small volume, lab scale screening of multiple candidates, and with the same instrument go to the scale up of the production for nano drug test and bioproduction. And so the current solution that exists is, you can make nanoparticles using conventional methods, talent injection or film-film hydration and extrusion, but that usually gives, I'm sorry for the format, I think this is Google slide changing, yeah, it's okay. So you usually have a broad size distribution. It's a multi-step process. It takes a lot of time to, it takes a lot of time, and you have a high variability from batch to batch. So this is something that people have been interested in microfluidics to solve these issues. So it's a very simple process. You have your lipids, you have your aqueous phase with your drug. You mix it in a mixer. You have many different kinds, and that usually leads to good narrow size distribution, a great control. It's a single-step process and highly repeatable. And so the solution that we propose at Insight Therapeutics is to have a new platform, a versatile lipid nanoparticle platform for the formulation. We can go from really low flow rates to high flow rates using the same platform. So you don't change technology to go across all these flow rates. And we also work, and this is what I work also during my thesis, we developed an innovative method to scale up the production from small flow rate to high flow rate while keeping the same size, the same quality attributes, the same size, the same PDI, and all the different characteristics. And so this is really under development. I will not go so much on this, but this is really the core of our technology. And we can produce highly reproducible LNPs of a wide range of sizes and very low PDIs. And one thing that is very important, I think, quite important for us is sustainability, because we all know that microfluidics has kind of a problem with the waste and the single-use cartridges. So we are really trying to include and to choose autoclavable. It's choosing actually the first word. Choosing autoclavable materials and develop cleaning protocols for reuse the materials, the chips, as much as we can, but also give the opportunity for the user to change the materials if they need really sterile conditions or really zero cross-contamination. And so why our solution has a chance to work, we really work to enhance the user experience. We know microfluidics can be sometimes tricky to handle. There is a learning curve for people who don't know microfluidics. So we really try to integrate that into a simple platform. This is the one that you see here, and maybe you had a chance to see it at our booth. So it's really under your control. We have this innovative mixing technique that we're currently in the process of patenting it, to scale up the production using the same method. And we have made multiple proof of concept using this technology, including one with real-time in-line size monitoring by DLS. And we made a poster on that. If you had a chance to see it, that's great, because it was really a great collaboration with Cordouan Technologies and UTCBS lab in Paris of Nathalie Minier, and we really could monitor inline and in real time the size of the particles. So that was really great. We already have a platform that is sold to different clients, and they use our technologies. They have good results. We have good feedback, and also feedback that enables us to improve ourselves and to propose some better products. And the proof of concept that we're at this stage. So on this current platform, it's basically we have a control unit. You have our all of these. Well, I cannot, but all of these is actually integrated in a smaller platform. We have the mixer, you have your samples that you load, and you produce nanoparticles, and we're able to test it on different formulation, quadsomes, so they are lipid and surfactant-based particles, PLG nanoparticles. We had a really good size control and fairly low PDI, so that was really promising. And we also did RNA encapsulation in liposomes, in LNPs, and had really good encapsulation efficiencies across all the different tested conditions, over around 95%. So that was really encouraging for us. And the next steps, we are currently working hard to release the platform as soon as possible. probably next semester. And then we'll work with, develop this patented mixer for, I was saying, using the same method from screening to large scale. And in the end, we're going to try to meet and certify it for the machine to be GMP compliant. Yes, and with that, I thank you very much for your attention. I will thank my team, Thomas, Alexis, Robin, and Mathieu, who are working hard with me to make that possible, and also Gwenelle, Elio, and Brahim. And please follow us on LinkedIn. If it's not done yet, you can scan the QR code. And yeah, if you have any questions, please shoot.
Thanks a lot, Audrey. Any question from the audience? Yes, Aikaterini. Very surprisingly.
Thanks. Yeah, thanks. Really, really, really interesting. I was just wondering, is distributed manufacturing, so manufacturing at the point of care, say, in hospitals, something you consider at all in your future?
Yes. This is one thing. We're currently also working with a partner in France, in Angers, who is interested in that, into having this platform, let's say, at the patient bed, or like in hospital pharmacies, I think that's the name, where they can readily produce the drug at the bed of the patient and go for more personalized medicine. So this is something that we're currently interested in, and we're looking for what is the the quality control that we need to put in place, what is all the certifications that we need to meet, and that's why I was very interested to know about like regulations, and we're really learning on how to make that possible using this system. So yeah, it's not yet done, but we're very interested in this.
Do you think there's regulatory hurdles? Because it's quite challenging, isn't it, from a regulatory point of view or something like that?
Yeah, so I think, in France, I think there is one thing that, a rule that changed actually that enabled pharmacy, like hospital pharmacy, to produce drugs in the hospitals. So that was not possible before, and on some specific drugs that are actually in shortages, there are a few formulations that are in shortage, either the API or just the capsule itself. So in that case, there is some, it's not loopholes, but they made some change in the law so that hospital pharmacies could actually produce the drug in the hospital and not go through the whole manufacturing and GMP manufacturing process. But yeah, we're really, that's why we're also busy with this collaboration to understand what is the regulatory framework. Yeah.
Very nice talk. I really like the symbols, how you summarize the duties for everybody, from the coins to shaking hands and so on. Yes. Maybe there are PhD students in the hall. I just wonder how your daily work changed from going from a PhD student in working in a spin-off company. Yeah, so actually for me, the transition was very smooth, because we started the company when I was still doing my PhD, so it was before the writing. So that was quite challenging, and I was also doing a PhD in an industrial environment. So that also helped. I also learned how, how the industry works. So I think that was in that way easier. But all in all, I think when we get out of a PhD, we're also super ready to learn and ready to meet new challenges and to adapt to new environment. And I think it can be tricky to go from academia to industry, but yeah, especially in startup, I think it's very dynamic. It's a place where you learn everything from scratch. So it's not a problem at all. Yeah.
Thanks a lot.
Yeah.
Have a nice and a safe trip back.
I will run.
And it was a pleasure to welcome you. And now, the person that has the world record of questions during this conference and of talks, Marco Monopoli from the Royal College of Surgeons in Ireland, will speak about diagnosis tools based on glycan detection with nanomaterials. Maybe some of you saw the presentation about the nanocarb Marie Curie project yesterday. Now we'll learn how to make money out of that.
Thanks a lot. So I have a plane to catch and not to Dublin. I'm not a great swimmer, so hopefully I will be on time. So we have I have a poster in case somebody will have more time. I'm just going to show you a brief overview of the work is being done by a PhD student from my team, Zin, and it was a co-fund project by the industry. So again, now we are stuck with the glycans. I just got very excited about the glycans. It's a post-translation modification that occurs pretty much everywhere. Most proteins are glycosylated, and the glycosylation here, they're just a type of the glycan type. And if you're not familiar with the glycan nomenclature, each glycan is, in this case, has a different color, and they are attached and they're trimmed by specific enzymes during the post-translation modification process. We now learned that the glycans can also be very useful biomarkers, and in several diseases, mostly chronic, and also during inflammation, these enzymes are either upregulated, downregulated, they can make mistakes. And now it's very well established that some specific glycosylation changes can be linked back with a particular disease. Because the glycosylation occur at a very early stage, then it's early warning diagnostic tool, because when you have higher level of proteins or protein mutation, it's a later stage of a generally later stage of the disease. So for this reason, they are quite attractive. So the N-glycans expression in the blood, they are quite stable. So the glycans, the proteins tend to keep the same protein structure in a very stable way, but they are very, very complex. So if you see here on the panel B, these are only the glycan structures of the IgG, and in this case, also changes in the glycosylation of IgG can lead to chronic disease like arthritis rheumatoidis and so on. So this is just some particular expression and profile of glycans from the IgG. So if you go back to the pool of plasma proteins, then it gets more complicated. And most importantly, you need a dedicated infrastructure to analyze the glycans. And I had the opportunity, when I was finished my postdoc, to spend a year in a company near Oxford, where I just spent 1 year just focusing on that. So vision and mission, I had a bunch of nanoparticles when I was in Dublin, and then when I was in UK, we were discussing with the company what we do with them. And basically what I offered to the company that was very attractive is that I could control very well the biomolecular, the proteins that we have on the nanoparticle surface. During my postdoc, I developed a lot of protocols to isolate, and in some way, we are starting to understand how you can control the physiochemical properties of the nanoparticles and fish out some protein from the mixture. So we came back from a problem to an opportunity. And I was not the first to do this. There were, by the time, there were a lot of groups with which I've been collaborated in the past, like Morteza Mahmoudi, Giulio Caracciolo, and so on, where they came up with the concept of the personalized corona. I think that it was very clever. And then during the years, they were looking for specific proteins pattern, and then they were able to track back with a particular disease. Now, going back for discovery biomarker, for those that work in discovery biomarker, you know that you have a big problem, which is the complexity of the plasma proteome, where you have about abundant proteins. They occupy nearly the 99%, 90 to 99% of the total proteome. And then within, so you have abundant proteins that really mask the signal, but then you have a very small percentage of pool where there are very attractive and interesting proteins that might tell you that something is going wrong. And these are what we call the tissue leakage, that they are generally detected if an antibody is really good or if the amount gets really high in blood, or a combination of both. So the solution, surprise, I just thought maybe the nanoparticles can help to clean up and enrich the signal. So what we did is that we came and we started profiling different type of nanoparticles, and then to see which pool we were isolating from the mixture. And because the plasma proteins are naturally glycosylated, then we were wanting to see if the glycans were accessible on the corona, and then I have to develop some protocols to pick them up and detect. And that's something that is worked successfully. It's been published, this paper, in ACS Nano quite recently. And then we thought this can be a very quick solution. The nanoparticles are very stable. They can also be shipped and shared with other groups. So it might be a tool, can be useful for other laboratories. And then the glycans can be also isolated, and we have this protocol. So we just go from 1,000 proteins from biological mixture to few hundreds isolated from the corona. And then it can be quite versatile because you can use all the nanoparticles that you want. So unique selling point, it's a well-established paradigm. The protocols are robust. They are reproducible, at least for the particles that we're using. Obviously, if you change nanoparticles, if you use soft material, then you need to go back and start again. But then again, if you change the surface properties of the nanomaterial, then you can come up with a different type of glycoprotein. And this is the validation, where we were looking, if you can see here on the left-hand side, this is a typical glycan analysis, glycan profile of the plasma protein. And then with the condition that we developed, we saw that the glycan profile was quite different. So each peak represents a different glycan type, a glycan structure. And then you can see here with the mass spectrometry, we're able to also to identify glycans that were enriched in the corona that were not picked up in the other system. If you can see here, these peaks are tiny, they're really, really subtle. So you, and then might mask a particular glycan structure that potentially can be affected during particular disease or condition. In this case, this is the proteome of the corona. So you can see here this is an SDS page of the plasma where you can see the serum albumin quite high. But in our corona, we saw a big enrichment of apolipoprotein A1, which is this protein at the bottom. And then we have a big signal that comes from fibrinogen. So we thought maybe fibrinogen might be it might be useful for some clinician just to be able to isolate fibrinogen with ease with these nanoparticles. And then also as a quality control, we compared the glyco profile of fibrinogen, purified proteins, and the one in the corona, and then we saw quite similar overlap. So it was validation that with this particular protocol, with these particular nanoparticles, we're able to isolate fibrinogen. We went one step further. This is a very small pilot, just to put a proof of concept. So there were 25 patients suffering from lung cancer, and then we have the match control, which were non-cancer patients. So there were persons in the clinic that didn't get the diagnosis, but maybe they have COPD, so other type of disease. So it was very tricky to dig down and look for the different variability. But then overall, as a first pilot studies, we have seen that there are some subtle but solid glycan structure changes. And this is just as a proof of concept. We had the opportunity to work with the clinician that were able to give us some samples to profile. And that was the glycan structure that we've seen that is being specific to lung cancer, and then will be validation on this biomarker to see if it's anything that can be exploited and used. So next step, we need to simplify even further the platform. Just going with the glycan analysis platform, you need a dedicated machine. Enzymes are very expensive, so we need to simplify as much as possible. The company was very happy, they're on board, and we're continuing this journey. So if you want to get rid of postdoc, just drop an email. We'll be happy to talk about it. Okay, thanks a lot.
You had 2 seconds left, so yeah, you could make something out of that. Yeah, I know. So then I can see. Yeah, if you want really to get rid of postdocs, you have now. Yeah, great talk. I loved it. First, questions from the audience, maybe. Yeah, Wafa.
Thank you very much for your talk. Just a very quick and simple question. How do you select the optimal nanoparticles to isolate, like the protein or marker of interest?
So that's an excellent question. And because this is an industry academia project, I have to be careful that you want to make sure that the industry is happy. But for industry, it was interesting. For them, it was a very low risk project because the students in my lab, the co-fund was not very high, so the cash contribution was not massive. So we actually had the opportunity to explore as many particles as possible. So it was a fishing expedition where you just try all the different combination, all the different protocols, and then once you go there, you see what glycoproteins you're picking up. And then you see which one is it might be of interest. So we just it's completely bottom up, where we just saw what we were taking out, and then we just started from there. Once we saw that we were able to pick up fibrinogen, then thankfully had some master students, and then we were getting this master student to see if we can actually optimize as much as possible. So this is not isolation, it's a prefractionation, where we have about 80% of fibrinogen-rich corona. So you have some background signal that comes from the other protein, but it was high enough to get a decent signal. Something we're very surprised is also the variability between the different donors, because people will have their differences. Maybe they have a lot of water the night before, or maybe they have the salty pizza. So that's something we wanted to check, how much variability there were. So in the paper, there is everything. There is all the SDS page. There were some difference in the protein, not massive. The fibrinogen band were very consistent. And it seems that the glycan are much more stable than the protein.
Keep this one. I have 3 of them. This one is going to...
Oh, sorry, I forgot the last slide. There is another one. How can I go? This is the lab. This is Zin. He's the PhD student that is now in the United States. And then again, there is a lot of people were also working in some way with the team. Sorry. And sorry, it's Ludger is the company. They was co-funding the project.
Thanks. So, yeah, very fascinating. Thanks. So I wanted to pick up on one thing you said, that one patient rarely have just one condition. They may have multiple conditions. So I was wondering, I guess these complicate things, and I'm wondering if there's some specific condition that is better to target with this approach than others, or.
Well, yeah, that's. I'm afraid it's not a question I can answer. We also did the same study with the CVD, cardiovascular disease, and we have a lot of data. In that case, we were looking for a person with a high risk, which means high blood pressure, cholesterol, and some patients were taking medications for cholesterol. Some people had a higher BMI than others. So I think, I don't know, I think we'll find out as we go along, but the trick is to work with the statisticians from the beginning, and then with the clinician also to see what they have, because at some stage it might be that the clinician is very excited, very interested, but then they say, we want to have a dedicated phlebotomist to pick up the sample. And then it might be that for a particular ward there is one, and then you just, you align with that particular cohort. So at the moment, I didn't have the luxury to say, let's pick up this disease. So I'm just engaging and see opportunities that are around. But that's a complication of discovery biomarkers. There are different factors. Also, the glycosylation changes also with age and also with the gender. So there are also other factors that have to be taken when we look for the AUC and when we just look for the stats.
Thanks a lot, Marco. I guess you can run now. Yeah. No, I love the concept, because simply to use something which is supposed to be a problem of nanomaterials, the corona, and make it a chance to make it a diagnosis out of that, that's great.
Let's hope.
Have a safe trip back. And now we are welcoming Cian McCrudden. So many changes in the program, sorry about that. Now it's up to date. If you look at the program right now, it's the right one. Yeah, after the conference, as you do. So Cian is coming from Queen's University of Belfast, exactly.
Correct.
And you, no, tell me the right one. No, please, come on.
Well, I am from Queen's University of Belfast, or I'm affiliated with it, but I work at Cian Therapeutics.
Even better, that's the translation of part of the presentation. And so he will talk about how to deliver without LNPs some vaccine against papillomavirus with RNA, right?
Correct.
Fantastic. You can take this one, it's the one. And you have the clicker that Marco left away. Oh, yeah, if you wish to see it. You're so demanding. Thank you.
Thank you very much for the
They will come back, Be patient, come on. It's day 4.
Yes, thank you very much for the introduction. Cian McCrudden, and yes, from Fion Therapeutics. Helen McCarthy sends her apologies. Unfortunately, she was double booked today, but I'm going to be able to give you a brief introduction to Fion Therapeutics and how we are using a peptide-based delivery system as an alternative to lipid nanoparticles. And a specific example I'll give is studies that we have done looking at a therapeutic vaccine for HPV-16 associated malignancies. So to give you a very brief introduction to Fion Therapeutics. So Fion spun out of Queen's University Belfast School of Pharmacy just over 6 years ago, and the work that we do at Fion Therapeutics is based on the technology that has been developed over the course of about 18 years that has been developed in the laboratory of Professor Helen McCarthy. And in the last 6 years at Fion Therapeutics, with a lot of support from a range of different people, sort of starting with our main investor in Vox Pharma, who are our lead investor. Also, a lot of support that has come from Innovate UK in the form of research grants, and in collaboration with a lot of other people. It's kind of a little bit obstructed here, but the ecosystem at Alderley Park, where we have received a lot of support in terms of intellectual property, CMC support, regulatory support, as well as the Medicines Discovery Catapult, and Sarah is here in the audience. They have provided support, and again, in terms of CMC support, the Centre for Process and Innovation has helped us get to a stage where we're going from purely academic research at Queen's to being at a stage where next year we hope to be able to, or we will be starting our first time in human clinical trial looking at the safety and tolerability of our HPV therapeutic vaccine. We have a number of different vaccines in our pipeline at Fion Therapeutics. Our V1 vaccine is the HPV vaccine, but we have a number of other vaccines that are that have oncological therapeutic vaccines for cancer conditions, and also prophylactic vaccines for infectious diseases such as COVID-19. We deliver most of our vaccine, our oncology vaccines, via intravenous route. We get really high delivery to antigen presenting cells via that route, and our prophylactic vaccines are via the intradermal or intramuscular conventional route of administration. In terms of how we produce our nanoparticles, so I know that Ying and Ahmed have both presented on RALA this morning, but very, very briefly, RALA is a positively charged peptide that will electrostatically interact with negatively charged messenger RNA. We use the Precision Nanosystems suite of microfluidic devices to produce in a very controlled manner, very predictable nanoparticles in terms of their size, their charge, and other characteristics. These particles have an overall net positive charge. These can be filtered, and then we run a suite of critical quality attribute analyses looking at the size of the nanoparticles to make sure that they are the size that we want them to be. We determine that the charge, or the nanoparticles that we produce, have a low PDI, so we know that it's a homogeneous population. The integrity of the mRNA inside the nanoparticles is really good. The process of producing the nanoparticles has a very, very low wastage, essentially no wastage. So the concentration of the nanoparticles in terms of the RNA, we get out what we put in, and we encapsulate overnight. We would say 100% of the mRNA that we put into them. You know, for wiggle room, we say more than 99%, but it's good. And we produce a product that is isotonic, so that when we administer it, we don't have any tonicity related issues. We produce nanoparticles that we then lyophilize. Usually, most of the assays that we do are use nanoparticles that are lyophilized. Lyophilizing particles has given us up to 18-month stability, and we have no reason to suspect that the stability stops at 18 months. Although we are also in the process of evaluating nanoparticles that don't go through the lyophilization stage, nanoparticles that are formulated and then the vials backfilled with nitrogen gas to prevent oxidation. And so we have some short-term stability on that as well, and that's going very, very well. In terms of how RAL nanoparticles work, we know that in vitro nanoparticles are endocytosed via clathrin-mediated pathways. They're taken up into the cells, and they, once taken into the endosome and the drop-in endosome pH that happens, the conformation of RAL moves from a random coil structure to a more alpha helical conformation, and that results in light fusion with the endosomal membrane and release of the mRNA cargo into the cytosol of the cells, with subsequent downstream translation of the protein. As I've said, we have a number of different vaccines that we are working on. In terms of our HPV specific vaccine, I'm not going to go into a lot of detail what's in this slide, but essentially HPV is a very, very well characterized virus, and we are very aware of its association with a number of different cancer conditions, and the E6 and E7 antigens are very, very highly characterized antigens. So these are the antigens that we incorporate. Our mRNA encoding these antigens is incorporated into our RAL nanoparticles for our PTX-V1 vaccine. We've done a lot of immunogenicity studies. This slide will not do it justice, but in a nutshell, we know that if we vaccinate mice in a two-dose regimen on day 0 and 21, and then cull the mice on day 42, we can take out the splenocytes from the mice, stimulate them with an E7 specific stimulus, and we know that they will produce a strong interferon gamma response. That's by a fluorospot or a variation on the ELISpot assay. When we use flow cytometry, we are able to determine that the cells that are responsible for the secretion of interferon gamma are CD8 positive T cells. We're not seeing a lot of activation of the CD4 positive T cells. So cytotoxic T cells are being stimulated using our vaccine. We have validated in vivo that the cells that we're provoking with the vaccine are able to produce a therapeutic effect. So what we have done is we have challenged C57 black mice with TC1. TC1 is a cell line that expresses the HPV E6 and E7 antigens. And after challenging them with the tumors, we then dose them with 3 doses of the vaccine and monitor tumor development or lack of tumor development in these mice over the course of about 100 plus days. The mice that do not have tumors, after 100 or so days, we would re-challenge them to see if there's any memory response to the cytotoxic T cells persist in the mice, sort of beyond challenge. And you can see that, bar one exception, there's always one, the mice that were challenged with the TC1 tumors did not develop syngeneic tumors after the first challenge or on re-challenge at day 123. So we obviously saw incredibly good survival. We used an alternative cell model. This is C343, which is another syngeneic tumor model, which again also expresses the HPV-16 antigens. It expresses the entire genome of HPV-16 actually, and we saw very, very similar response. Our vehicles developed tumors, and the vaccinated mice did not develop tumors. Something that we did with these mice at the end of the study, rather than just leaving it, we decided that we would take out the spleens from the mice. We would stimulate them with overlapping peptides from E7 again, and again in a fluorospot assay, we can see that despite 200 days having passed since vaccination, we were still seeing presence of antigen-responsive cells in the spleens of the mice. So the cytotoxic T cells that we're stimulating are persisting sort of very, very long after. 200 days is a long time in the life of a mouse. And when we analyzed the cells by flow cytometry, we found that there was a very, very high interferon gamma and TNF alpha response in the T effector memory cells in the CD8 population. So we have as I say, this does not do it justice. We have a lot of data to back up, to support our decision to proceed to clinical trial. The one step in the process of proceeding to clinical trial, we needed to undertake a pivotal safety and tox study, which was done at Charles River Laboratories in France. In essence, what that study looked like was we used rats. We delivered what we think will be the sort of the highest clinical dose, which is 200 micrograms of RNA, and we dosed those rats with a sort of an N+1 regimen. So if we're going to give 3 doses to a patient, which is what we think that we'll do, so we give 4 doses in the rats. We analysed the rats for a lot of different markers. This again does not do anything like just the justice that it deserves. The rats produced an immune response, both antibody mediated and cellular mediated. We saw very, very little or no observations that would indicate any concern. The body weight of the mice, the temperature of the rats. If I say mice, I mean rats. The body weight of the rats, the temperature of the rats, the food consumption, etc., etc., etc. No concerns. We looked. I'm going to move this out of the way. We looked at a range of different circulating biochemical markers to look at for toxicity of the kidney, liver, the blood cell counts, etc., and there were no concerns with those. And on gross pathology, there was a couple of maybe unexpected observations that were seen in the odd rat, but if we saw those observations in the rats that were culled on day 24, those observations had disappeared by day 43. So if there was something that happened that was observed 2 days after administration of the vaccine, it had rectified itself by 3 weeks later. So nothing of any concern, nothing that would prevent us from moving on to our clinical study, except the technology failing. And the timing, okay. Very quickly, this is a snapshot of what our clinical trial will look like. It's a modular design. We will start off looking at the safety and tolerability of our vaccine in HPV-positive advanced cancers. We will then, provided there's no issues, move on to Module 2. Module 2 will be in a SIN-positive, HPV-positive population who have, for whom we think that this would be a really very good treatment regimen or treatment modality. And then again, provided we have positive outcomes from that, we'll move on to Module 3 and Module 4, which will be in more advanced disease, looking at cervical cancer patients and head and neck cancer patients. This is the team. Sorry if I ran over. But thank you for listening.
Very stimulating. Any questions from the crowd? Yes. Oh, 3 of them. Yeah, you have Katerina's mic. You just need to unmute it to this like this.
Terrific presentation. A lot of information, a lot of data. It was difficult keeping up with you, but it looked fascinating. One of my questions is, anytime we introduce a protein or a peptide, we always risk an immune response. Of course, you desire that against your true antigen. I think, was it E7?
E6 and E7.
But then, and you do obtain a response, but have you looked at whether or not your carrier, RALA, I think, is immunogenic in itself? And if you would eventually, because you said you're getting 3 or 4 doses, you expect to, would you get the anti-drug antibody response?
Okay, so that is a really good question. We have done some work on that. There was some work that we published a few years ago where we were using RALA to deliver plasmid DNA. The nanoparticles are very similar in terms of their size, shape, charge, etc. So we have looked in C57s that have received multiple administrations of RALA plasmid DNA nanoparticles, and we've looked specifically for neutralizing antibodies. The way we've done that is we've immobilized nanoparticles on the well of an ELISA dish and then probed that with the serum of the mice that received multiple administrations of the nanoparticles, and we have failed to demonstrate any neutralizing or any binding antibodies. So we've looked for it and we haven't found it. So we don't think that it exists. We have also looked at the circulating markers of sort of an innate immune response, an undesirable immune response, such as IL-5, IL-6. And we have seen small increases in the amount of circulating IL-5, IL-6, but very incredibly small, like 2 orders of magnitude lower than seen in similar studies that were done using a lipid nanoparticle that was published by Genentech.
Hello, thank you very much for the really nice talk. I really enjoy it. So I have some question around your starting material, the polypeptides. So one question is about the cost of the raw material, because we know when you synthesize polypeptides, you usually do it in small quantity, and quite often, especially for nanoparticles, you require polypeptide to be very monodispersed in terms of their molecular weight and everything. So does that put a high requirement for your raw material, and does it then contribute to the overall cost of your vaccine product?
So again, that's probably a good question. I'm coming at this more from the biological end than the manufacturing end. What I can say is that we source our peptide from Bachem, who are able to produce it to GMP standards. I think I don't know about the cost of a peptide raw material versus the cost of for example, a lipid raw material. What I can say is that the patent attorney wouldn't like me to say that it's a simple manufacturing process, but it's a relatively straightforward manufacturing process that does not require the use of techniques such as tangential flow filtration, which is associated with lipid nanoparticles. So there's the process is a lot more streamlined for the production of the RALA mRNA nanoparticles than it is my understanding of the formulation of mRNAs in a lipid formulation.
Yeah, great. Sorry, just one.
Thank you for the presentation.
We can talk afterwards if you want.
Yeah, very nice presentation. I just want to ask, like, for the RALA peptide in comparison to LMPs, does it provide like more potent transfection? Have you compared RALA with LMPs, for example, in terms of potency?
Yeah. So this is a question that we're asked quite often, and that is actually something that we are undertaking a study on at the minute, where we're doing a side-by-side study looking at the immune response using nanoparticles that we're producing using the Precision Nanosystems GenVoy lipid product. So I don't have the data on that, but that's something that we're working on. It's a good question.
Welcome, Aikaterini Lalatsa from University of Strathclyde, which is exactly the most complicated word to pronounce for a French guy. And you will.
My name is so complicated.
It is complicated. You will talk about the fibers for brain therapy, right?
Exactly.
You have 10 to 12 minutes, as much as you want.
No worries. Thank you very much. I'll just use the computer. Okay, so what I want to talk to you about, and what is actually our vision for our technology that we call G-Fibers, is essentially we're trying to solve a simple problem. 1 in 3 suffer from brain disease sitting in this room or in the world in general. So we want to develop non-invasive formulations for treating brain diseases, and particularly we're targeting the nose-to-brain route to deliver this vision. So what we're hoping to achieve is essentially scalable, continuous processes to deliver nano-enabled solutions for proteins and peptides, but also for small molecules and other biologicals that can be administered by patients and can actually lead to targeted therapy in the central nervous system. So today I want to focus in one of these brain areas and diseases, which is particularly glioblastoma multiforme. So it's a brain tumor, it's the most aggressive brain tumor, and only in the UK we have every year 12,000 new cases. Now, why focus on glioblastoma? Because practically anybody that gets this diagnosis is practically a diagnosis of death, because they know that practically less than 1 in 10 survive after 5 years, and less than 1 in 4 will survive more than 2 years. So actually, we need therapies that can actually extend that lifespan significantly. Now, what do patients receive at the moment? What is the standard of care? So it receives a triple protocol which involves radiotherapy, chemotherapy, and of course, initially, surgical debunking, so removal of the tumor from the brain, and essentially allowing these treatments to work together. But even with standard of care, typically, we don't go above a 15 months survival. So that is very abysmal at this point of time. So you actually need something that can actually take us further. So what are the 4 main problems and why we have this survival? The first one is essentially the presence of the blood-brain barrier, which is a physiological barrier that doesn't allow actually, almost near percent of our drug, 98% of the drugs and almost all biologicals from entering the brain. The second issue is essentially resistance. So temozolomide, that is the chemotherapy of choice, actually resistance has been shown quite readily. There is radiotherapy resistance that is also attributing to that, and a lot of the patients develop metastasis quite early, and that can lead essentially to death of patient. So in all of these cases, these approaches are non-targeted, so they actually attack healthy cells in the brain as well as the tumor cells. And the final one, essentially, over the years, we've tried to deliver a local way of treating brain tumors. So when the surgeon goes in and essentially eliminates the tumor mass, we try to use wafers loaded in carmustine or with paclitaxel and try to allow for local release of these essentially chemotherapeutics to eliminate essentially the tumor cells. However, that only works for a limited period of time, for 30 days, and only really diffuses around 5 millimeters from where the surgeon has excised the tumor. Any other treatments that are in development? Lately, there is a company called CNS Pharmaceuticals that is they are claiming that they have the blood-brain barrier permeable alkylating agent called berubicin. They are trying to go into phase 1 trials with these alkylating agents. So we need to see how that will pan out over time. There was an antibody that was tested a bit in the last decade, but actually in phase 3 studies, it actually failed to demonstrate significant benefit to the patient. There is a glycolysis inhibitor, D-chloroacetate, that is actually currently in development, but again, toxicity issues might become problematic. And the only other therapy that actually has been approved by FDA is the use of these alternating field caps, which the patient has to wear all the time in order to get any benefit, and really does not extend the survival more than 20 months. So this is what currently is actually available. So why do we think that we have a better chance than all of these people? So we know from patient biopsies that around 40% of patients overexpress a G protein coupled receptor. This is a receptor family that is highly druggable, and we know that express a protein receptor, the gonadotropin-releasing hormone receptor. And we know that patients that have been treated with a commercially available analog of this peptide, they've actually shown a dose-dependent anti-proliferative effects, and the tumor can actually be treated. Now, why the strategy is targeted and specific is because the mechanism of action of this hormone in cancer cells is different to the mechanism of action in hormone cells, like pituitary cells or other hormone sex cells that we have in the body. The problem of using the commercial formulation, Zoladex, is that this is a PLGA implant that has a long shelf life, but actually has a very short half-life in the blood, and again, the hormone does not cross the blood-brain barrier. So this is where we are actually coming into the equation. Our technology is simple, is based on a site-specific lipidization process that you can actually amend to this hormone, but also to other peptides. So when we actually create these amphiphilic peptides, we can actually do 2 things. First and most importantly, we maintain the receptor binding, so we do not alter the specific activity of the natural hormone. And secondly, we trigger its self-assembly. So the peptide now is not only the active moiety, but is actually a drug carrier, that can also carry other molecules that are brain impermeable to the brain. And what we've actually shown is that we can actually produce these nanofibers that are typically around 800 nanometers in length and around 20 to 40 nanometers in width. And we can actually use them for a lot of purposes. When we actually have these amphiphiles, we've actually shown that they have a very good biological stability, both in plasma, in brain, and in liver homogenates. And you can also load in these fibers clinically relevant concentrations of brain impermeable alkylating agents or other cytotoxic agents. And one of the molecules that we've shown here is paclitaxel, which essentially typically is used as a cremophore formulation that is infused, that has a lot of side effects and needs to be infused slowly to avoid side effects, and does not cross the blood-brain barrier. We can actually entrap concentrations up to 2 milligrams per ml of paclitaxel, and we can actually allow its solubilization in aqueous environment. We've actually had a formulation that is already available as an intravenous formulation that is lyophilized, and we have stability data for 2 years. And we know that manufacturing these peptides is possible in GMP level and large scale without a lot of issues in obtaining these compounds. We've tested our peptide in different types of cell lines. Particularly, we used U87, but also patient-derived biopsy cell lines. And we've actually seen that we have activity in the micromolar range, both in normoxia and in hypoxia. The peptide alone can cause G2M phase arrest, and actually that is potentiated when it's actually used together with paclitaxel. It can actually cause early and late phase apoptosis, and it activates a specific pathway that we're hoping to reach. And we have specific binding to this G protein coupling receptor, which we've actually shown with single molecular force spectroscopy studies that we've done in collaboration with the University of Linz. We've done some preliminary pharmacokinetic studies, and we've actually seen that we can deliver approximately 0.5% of the injected dose to the brain. And we are hoping that that will be potentiated by targeting the peptide via nose-to-brain route. And at the moment, Karim, my PhD student that's in the back of the room, has actually presented a poster here in the conference to actually show some of the work that we've done with microfluidics to allow for a continuous formulation process generation that, when it's coupled with atomization techniques, can allow us to produce nano in microparticles for nose-to-brain delivery of the fibers. So where we are at the moment, we are actually trying to go through a competition to create our own university spin-out company that will actually have 2 patents associated to that. One, of course, that we've already filed for the fibers, and one that we're in the process of filing essentially for the carbohydrate polymers that we're using for nose-to-brain delivery. And essentially what we are actually looking, and if this is something that is in interest, we're actually looking for partners that already have their molecules or nanoparticles that they would like to actually deliver non-invasively for brain indications, for a range of indications that we can actually co-formulate and develop together towards essentially getting these medicines essentially to patients. So I think I will leave you with that. This is some of my partners. I want to especially give thanks to Dr. Leite, which was one of the first person working on this project and has done significant work. Dr. San, Dr. Figueiredo, Dr. Baquero, and of course Karim that is in the audience that is actually working on the nose formulations and the microfluidic process for continuous manufacture. And of course everybody else that has helped me in the journey. Thank you for your attention.
Busiest slide, but shortest presentation.
You see?
What a show, what a performance, Aikaterini.
Thank you, Aikaterini, for this very inspiring actually results. I mean, you mentioned in one of your results a 30-fold increase in brain permeation.
Yes.
This is incredible. Okay, usually people are spinning out or patent only with
much lower percentages.
Exactly. Yeah. So this is great. Why you promote the GBM and not other type of diseases?
We actually have data on 2 types of diseases at the moment. One of them is GBM, and the other one is in the area of stroke.
Okay.
But that is a completely different peptide, G protein coupling receptor peptide targeted molecule, that a similar strategy has been applied, and we've actually seen quite good data in neuroprotection, and that's in the field of stroke.
Can I do one small suggestion? Okay.
Yes.
So Innovate UK, at least, sorry, it's only for the UK people, has a program from Set Square that it's called IQ Discovery, which means you have to ask 100 clinicians, and they will guide you where your solution is
would
going
be ideally
to be
suited.
applied, because it will be a little bit problematic if you go with 2 areas, 2 therapeutic areas, and not to investors or afterwards for grants, etc. So it's better to pick up the most suitable for your solution.
The reason why, at least from my side, that I've chosen these areas is that there is not actually at the moment any treatment for stroke, apart from the tissue plasminogen. So there isn't really any therapies there to compete against. Yeah, and the reason in terms of glioblastoma, yes, there are more therapies, but the need is still there. Yeah, and I think we already know that.
Was it a technology-driven or funding-driven choice?
I think technology-driven, I would probably say, because this is something that I have worked in for a number of years. I'm quite passionate advocate of non-invasive brain delivery, and for me it was something that I always considered as a possibility. I believe in the technology, so I'd probably say this is technology-driven, not necessarily funding-driven from that point of view. However, seeing the technologies that are available, I think that that can solve a number of issues as is. So that's why I think that if we spin this out, I see the company as two areas. One of them is based on the peptide modification, but another one that probably will be more mainstream will actually use the nano and micro particles for nose-to-brain delivery of existing formulations of therapeutics that other companies are developing at the moment.
And basically with some kind of platform IP management that you can
Exactly.
make, right? So being rich and then do you think the thing you like.
Exactly.
Fantastic, right?
This is
The
like
life. Any last question before we close? Yes.
Yes.
Thank you very much, Katrina. So two quick questions. First, you are talking about docetaxel or
Paclitaxel, yeah.
which is very hydrophobic, and tolozamide is basically the standard of care for, and this is a small hydrophilic molecule. Do you think you can deliver this molecule using your
Temozolomide.
peptide? Yeah.
I had this question before. I've actually tried to load it. I think the problem with temozolomide, I've managed to load it up to specific concentration, but much lower to what is actually currently administered. So when we've actually tested them together, I did get good therapeutic responses, so it is a possibility. But I don't think for this technology is the ideal, essentially, cargo. I have tried it with other alkylating agents, so I have used etoposide, I have used doxorubicin in the past, and we've actually had good data. So I think in the case of temozolomide is the mass to peptide ratio. The temozolomide in such higher amounts compared to the peptides that actually I'm not getting the good level of encapsulation or the level of encapsulation. So I need to use increasing amount of peptides to actually manage to achieve that. So that wouldn't be my ideal, let's say, formulation to carry forward.
And the second question related to all nanotubes stuff. You monitor the diameter, but can you really also control the length, which is a bit tricky with these?
I think this is something that we've noticed with microfluidics, that actually when the preparation is done microfluidically, and this is something that there isn't a lot of data actually on microfluidics and long axial particles, we can actually control the length, and I think also the, essentially the size of the nanofibers. So there has been some work, I think with a beta in microfluidics, that they have actually shown that depending on the conditions of manufacture, you can actually control the length of the fiber that you are actually getting. And that is based on the flow rate and on the actual architecture of the microfluidic chip that you are actually using. So I think that is possible, but it's not something that I have definitely controlled, let's say, in the initial experiments that we have done. The good thing about the size is that we didn't have, when we injected them intravenously, we didn't observe acute effects from lung uptake or high levels, in a sense, in the liver and in the lung that actually would cause a trigger in terms of toxicology from that point of view. Of course.
Thank you so much. That was a brilliant session. I like it very much. Thanks to the speakers, and please, so beware. Posters, take them down before 2. Enjoy lunch until 1:30, and I have the names of people escaping right now. Stay for the awards, please. Thank you so much to the speakers. That was great.